So this clicker doesn't work. So a little bit about what I did before college. I was really into music. Also, here I am destroying a birthday cake sometime in the 90s. But I'm really into music. I was the drum major for my high school band. And I led the band in parades and field shows during the halftime of football games, things like that. Played guitar. I played in a band for a little bit. I played bass. And a little thing I do that's a little uncommon is I solve Rubik's Cubes and collect them. So I'm going to do that at the end. I had an assistant mix it up, so I'm not cheating. But yeah. I'm from Kansas. It's the middle of the country, really far away. Not as far away as some of you folks, but yeah. The flag of Kansas is really similar to the flag of Oregon. It's one of those where it's just a blue background and the emblem and the name. It didn't originally have the name. It used to be less descriptive than it is now, but I think I like the Oregon one better because it has a beaver on the back, but I still like it. And whenever I tell anybody I'm from Kansas, they immediately go to the Wizard of Oz. They'll ask me something like, "Where's Toto?" or "There's no place like home, you're not in Kansas anymore." And I've talked to a bunch of my Kansas friends, and we've all decided that it's so ubiquitous that everybody gets one. Like you can say it once, and then after that it gets old. But yeah, a few facts from Kansas. Became a territory in 1854, became a state in 1861. And that period of time was called Bleeding Kansas because there was a lot of political turmoil in the lead up to the Civil War that I'll talk a little bit about later. But it was an early battleground. It has a lot of nicknames. Officially, it's called the Sunflower State, hence the sunflower. But more appropriately, it's called the Wheat State because it produces about a fifth of all the wheat used in the nation every year. And even more impressively, it produces about 60% of all the sorghum supply per year. It's a truly a breadbasket state. Famously, the explorer Zebulon Pike, who Pike's Peak is named after, when he came across the region that became Kansas, he called it the Great American Desert. And it's actually not a desert. It's just a prairie. It's actually somewhat moist, humid, subtropical, and it makes you wonder what he thought when he got further west and actually saw some desert. But the flora of Kansas is, like I said, prairie, although it gets a little more woodland to the east. But it's most famous for the tall grass prairie of the mid-east of the state. Here's an example. This is the Kanza Prairie. It's beautiful, but unfortunately, it's kind of the last one. This and a few other little preserves are the last vestiges of the tall grass prairie, because most of it looks like this now, like crisscrossed and just completely dissected by roads and development, agriculture. And it's a really endangered ecotype, the tall grass prairie. What's next? Oh, this is not exactly a tall grass prairie, but this is kind of the most preserved natural area of Kansas. It's called the Flint Hills, or the Osage. And it was preserved because you can't really grow anything on it. The bedrock is right at the surface, and the soil is really thin. You put a plow in it, you break your plow. So it's really only used for rangeland, which I know means it's not exactly a natural environment, but it is still beautiful. You can go there and look all the way to the horizon, not see a house. You just see trees sort of in the riverine channels. But this is where the bison used to roam. Now it's just Eurasian cattle, not bison. I actually don't think there are any bison in Kansas anymore. Maybe in some preserves. Oh, here's another picture. It's really gorgeous. When you drive across Kansas, there's long stretches like this, and it's big sky country for sure.
So is that mixed grass?
Yes, yeah, I believe so. Yeah, you don't get all the tall, like up to your waist stuff, and although they do burn it. So yeah, I think it's more mixed than the tall grass. So one thing about Kansas is there's crazy weather, something to do with the way moisture ramps up over the Rockies, creates a lot of thunderstorms during the spring and fall and during the summer. And when they're strong enough, they can produce tornadoes. And tornadoes cause a lot of damage each year. I couldn't find like super hard numbers about it, but it seems to be about 100 people a year die from tornadoes, which really isn't that many people dying per se. Like, I think more people die from having vending machines fall on them. but they cause a lot of property damage, like $500 million each year in property damage, and that's like ranch houses being destroyed, cattle being killed, things like that. And it's a really extreme weather event. They can range from like an EF0, they're rated on the enhanced Fujita scale. An EF0 is like a dust devil that you might see around here, and then up to an EF5. The fastest wind speed ever recorded on Earth was in a tornado in Oklahoma, which was about 300 miles per hour, and yeah, seriously strong wind. It's actually rotating, so it does more damage than just direct forced wind. So, yeah.
Have you ever seen any up close?
Yeah, I've seen a tornado before. I missed this one. So this one came by my hometown a month ago as part of the May tornado outbreak. About 300 tornadoes touched down in the nation, like in the country. I think 100 of those were in Kansas area. And this one went right by, like I used to drive down that road all the time. I know exactly where that is. That was a mile-wide tornado that traveled 30 miles and caused EF4, I believe, caused a lot of damage. Oh yeah? Yes, who?
Yeah, I have a question.
Uh-huh.
So I want to take some pictures for tornadoes.
Uh-huh.
So how close I can go?
Oh, you can get real close.
Real close?
Yeah, you can look up on YouTube. There's lots of like professional storm chasers, and they'll like find the road that'll get them the closest, and sometimes it passes right over them. And depending on the strength, like you could be toast, you could be safe, who knows? But if you build a house in Kansas, be sure to have a basement. You see, that's the only thing left of that house. They just get wiped. And this is a big nursery near Linwood, Kansas. I used to go here when I worked for a greenhouse to get exotic plants, and it just got demolished. I haven't heard much about it since then. I can only presume it's nearly a total loss aside from the property. I think so, yeah. It's pretty much necessary, depending on where you live, but yeah. Big cities usually don't get hit because if they got hit, they wouldn't become big cities, but that's not really a rule, it's just kind of a tendency. Yeah, so here's a map of Kansas. You can see the big city. The biggest city fully in Kansas is Wichita, and then this metropolitan area, Kansas City, most of it's actually in Missouri, and most of the other parts in Kansas are like split up into little subdivisions and stuff, so it doesn't really count as the biggest city. Capital's Topeka, right there. I'm from Baldwin City, that imperceptible dot there. It's a town about 5,000 people, nice quaint town built around a Methodist university, Baker University. Brick roads, it's a great place to grow up. Every year there's what's called the Maple Leaf Festival, where vendors from all around come and sell their wares, stuff like pottery, food, yard art, just all sorts of stuff. The smells are really memorable. It's like maple syrup and honey and caramel apples. Everybody's eating turkey legs. There are fairground rides. Such a great small town thing. Oh, and I found this is actually the same road, just looking a different way, High Street. It's interesting to see probably the early 1900s. I've ridden my bike down this road countless times. It's just interesting to see how long it's been there. But after I graduated high school, oh, also, on a side, the Maple Leaf Festival, there's a parade. That's what I was marching in earlier on. Yeah. So I moved really far away after high school to Lawrence, Kansas, about 15 minutes away to attend the University of Kansas. A little aside now to talk kind of about Kansas history. When Kansas became a territory, before Kansas became a territory, what was called the Missouri Compromise was in place. So this bottom border of Missouri, that's the 3630 parallel. The Missouri Compromise stated that any new states formed north of this line would be free states, and any states beneath it would be slave states. But leading up to the Civil War, there was so much political turmoil, congressmen from the southern states couldn't live with this compromise anymore. So when the Great Plains territories became territories, they passed what's called the Kansas-Nebraska Act, which ruled that any new states formed from these territories would be either slave or free states based on popular sovereignty. So people living there would vote if they would be a free or a slave state. And since there weren't a lot of people living there, what this caused was kind of a political land grab. So people from New England, especially Massachusetts, abolitionists who didn't want a new slave state formed in the Union, started converging on eastern Kansas, and then slavers from the southern states did the same, especially from Missouri. People called the border ruffians of Missouri would cross the Missouri River and raid. It's essentially guerrilla warfare. They wanted to do a few things. They wanted to settle if they could. They wanted to kill abolitionists if they could, and they wanted to destroy abolitionist stuff to scare them away because they wanted another slave state. And this is what's called the sacking of Lawrence. William Quantrill brought 300 men to basically the unguarded city of Lawrence and destroyed everything. This is the Eldridge Hotel, which has since been rebuilt. The street's called Massachusetts Street. It's the main drag of central Lawrence. And this building is actually still here. It still says dry goods and groceries, which is kind of amazing. But eventually the abolitionists won. In 1861, Kansas was admitted as the 34th state, a free state. Slavery was not allowed. And the Missouri-Kansas rivalry still proceeds to today. And after the Civil War, things kind of calmed down in Kansas, and people of Lawrence decided to build a university up on this hill. It's called Mount Oread. It's not really a mountain. It's like 300, 400 feet above the prairie floor, but it's Kansas, come on. So yeah, that's the University of Kansas. That's where I went to school. Their mascot is the Jayhawk. Jayhawkers were a nickname for the abolitionists in Kansas who fought the border ruffians. And KU is actually famous for basketball. If you follow NCAA basketball, they do very well most of the time. And that's kind of a tradition because James Naismith, the inventor of basketball, formed the first collegiate basketball team at KU. And actually the original handwritten rules of basketball are housed, you can hardly see it, this building here is Naismith Hall, home of Allen Fieldhouse, where basketball is played. And yeah, we have the original rules of basketball written by the creator. And so while I was there, I worked for the greenhouse. Freshman year, I got a job, great job, just working as the greenhouse assistant to Katie Sadler. And we took care of, I think there were 6 greenhouses while I was there that we took care of. And then any plants that faculty were doing experiments on, we would maintain. And also anyone with an office plant that looked sick, they would take it to us, and we would usually just prune it and repot it. But kind of like plant doctors, not as much as the clinic is here. But a fun thing that I did is we worked on green walls. I helped install this one, which is like a vertical, this one's like 16 feet tall. It's hard to see with the perspective. It's like 16 by whatever, 30 feet. And it's all ferns. It's all ferns and bromeliads and hydroponically watered from the top. There's a drip line that goes through, and they're planted in like a root ball into a felt backing. And that was really fun. I helped install that one, and we maintained that one and the one she's in front of here, which is in a different building, for however many years. I think that one was 3 years, and this one was all 4. And then in my last year of university, I worked for Dean's Professor Joy Ward, a brilliant ecology evolutionary biologist whose our work mostly focused around how differences in atmospheric CO2 confer differences to plant development and ecology. Because if you think about it, CO2 is kind of the main food for plants in the air. It's the carbon source. And when plants have different strategies of acquiring carbon from the air, they're going to be differentially affected by changes in CO2. So if you increase CO2 on a C3 plant, it'll just grow more. It'll be able to fix more carbon, make more plant. whereas a C4 plant like grass, wheat, corn is not going to be as is not going to be as successful. It's not going to be able to take advantage of the extra carbon in the air because C4 plants concentrate carbon already around rubisco. So implications of this are huge in ecological settings because it means just the population dynamics of what's going on is going to change. Plants that can take more advantage are going to get more populous. Plants that can't won't. And in an agricultural setting, lots of weeds are dicots, C3 dicots, and lots of crop plants are C4 monocots that aren't going to be as advantageous. But when I worked for the lab, I assisted basically all these people in just day-to-day activities in the lab. It was a good undergrad research job. But when I graduated, that's my mom. When I graduated, I really was interested in biotechnology. I graduated with a bachelor's in biochemistry. That's me. Graduated with a bachelor's in biochemistry, and I was really interested in biotechnology. And I got in contact with Emmerich here, and he had a NIFA fellowship that he was willing to give me to come to study here. And so I did. And I accepted it. And here, time-wise, this is May 2017. And if you'll remember, August 2017, there was an eclipse that passed right over Corvallis and where I used to live. So I waited till the 21st of August when this happened. And me and my friend went like a few miles north to be right under totality, and it promptly started to rain. It got cloudy, and this is the best picture I got. You can sort of see. But it was a great experience because we were able to get under totality. And when this was happening, it was really cloudy, really eerie. This is like 11 o'clock in the morning. And so all around, it looked like the sunset was happening. It was really eerie. Crickets were coming out. They thought it was nighttime. Nocturnal crepuscular animals were just completely confused what was going on. But I'll never forget that. And then the next day after this, I did a huge journey from Lawrence to Corvallis. It took I didn't do it in one day, obviously, but yeah. So here are some totally safe and legal pictures I took while I was driving. This one's in Kansas, nice prairie.
It looks like you were parked.
Yeah, no, this is in Wyoming, just outside Cheyenne. It's a national park, I can't remember what it's called, or maybe a state park. These are the Tetons, clearly, and just completely gorgeous, yeah. It looks like Bob Ross's signature is gonna be in the corner just of the horizon. It just doesn't even look real. This is a volcanic butte in the Snake River Valley in Idaho, right outside of Atomic City. There's an old, one of the first nuclear reactors near there that we stopped. It was interesting. And then here, yeah, got to Oregon, entered in Ontario, and I've been there ever since. I did my first year in Corvallis, main campus, getting my coursework done. And then in June, last year in June, so I've been here a year, I moved up to Herrick, up to Hermiston. And so now I'm gonna actually talk about what I do. I don't wanna bore you too much with things that you already know, like how potatoes are a starchy tuber-bearing member of the Solanaceae family. And they're very important to agriculture, just worldwide, and especially to here. Half of all the potatoes made, maybe more than half of all the potatoes made in the US are made in Oregon, Washington, and Idaho. And one problem facing potato production is potato virus Y. This is a virus that's transmitted by aphids, and really the only good way to control is with clean seed potatoes. And this means that seed certification is our first line of defense against potato virus Y. And seed certification is becoming more complicated due to the change in the strains that are happening over time. So originally, PVY-O strain was the most common, but now it's being replaced by recombinant strains, especially Inwilga, NTN, NO. Symptoms of PVY, kind of typical of virus symptoms. You can get chlorosis mottling, wrinkling of the leaves, vein necrosis, generalized necrosis, and these symptoms will cause a reduction of yield, clearly, but what's troublesome is that in recent years, strains that can cause the potato necrotic ring spot disease, rather, that will render a tuber completely unmarketable. And that's a huge problem. You want to be able to market your potatoes, clearly. And also, an economist from Idaho has estimated that about $34 million are lost to just the state of Idaho alone based on PVY damage. And there are losses of about 100 jobs as well. Yeah, it's a serious problem. And so many varieties of potato have resistance genes for potato virus Y. Most common among them are N genes. These are strain-specific resistance genes. If a potato has an N gene, then an N gene to a specific strain, when that strain of virus enters the plant cell, it can recognize it, and it initiates a cascade of events that cause these necrotic spots to appear, localized necrosis, programmed cell death, and the idea is that it walls off the plant from the virus. But the problem is it doesn't always work. And even if a plant creates these HR, this is called the hypersensitive response. That's important. You should know that. Even if a plant initiates a hypersensitive response because it has a resistance gene for a virus, that doesn't mean it's going to not develop a systemic infection. And so we need to better understand the resistance response. And that's basically kind of setting the stage for what I'm doing here. Before I was here, Amaric and actually Jim, who's sitting right here, set up an experiment where they were going to compare Premier russet, which has an N gene, to one strain, actually two, but the one strain that we're talking about, PVYO. It resists PVYO and not most other strains, and russet Burbank doesn't resist any PVY strains. And so what we wanted to do, or what they wanted to do, is see, aside from the resistance gene, which genes are differentially controlled in a potato variety that is resisting a virus. The way to do that is with transcriptomics. This is kind of a little more upper level biology if you're in high school, but this is, you have the genome that has all your genes in it. You can sequence that and see what genes are in any organism. But what genes are being used are transcribed. They're expressed into a different nucleic acid similar to DNA called RNA. And if you sequence that, you can see which genes are being used. And you can count how many of these of a specific gene that you sequence in one treatment versus the other, and you can see differentially which genes are being used differently. I'm going to say differentially a bunch. Okay, so generally what we see is three specific reactions. So when you infect Premier with a virus that it resists, there's a certain network of genes that are activated or deactivated. When you inoculate it with a strain that it doesn't resist, there's a certain network of genes. And then the variety that doesn't resist anything, you inoculate it with a virus that it doesn't resist, there's a certain network. So what we're looking for is genes that in Premier russet, while it's resisting a virus, are differentially controlled, like uniquely in this reaction versus the other ones where the virus is not resisted or is not recognized by the resistance gene. And so a number of genes were identified as candidate genes, and my project focuses on two. One of them we call ABC or PIP, and it appears to be a peptide hormone that that family of genes in other plants was seen to be involved in propagating a recognition of pathogens. So that makes sense that it might be involved in pathogen recognition. And then the other gene I'm looking at is called MYC. It's a transcription factor that is involved with wound and pathogen response. So we think if these genes which I see here, we think that these genes play a role in the hypersensitive response, or at least the resistance response to potato virus Y when the host has a resistance gene for a certain strain that it's challenged with. And so building from that, we think that modulating the expression of these genes will modulate the resistance to potato virus Y, or at least the response to inoculation in terms of the timing, severity of symptoms, whether or not a systemic infection develops. And so to do that, first line of defense is to take the sequence data we get for these genes. They haven't been studied in potato, but they might have been studied in other plants. They have, spoiler alert. But we can take the sequence data and compare it in databases to things that have also been studied. And from that, we can get an idea of what these genes might do. And that's how and then from that, from the sequence data, we develop transgenic plants that either overexpress the gene, that is, have it on all the time, versus normally genes are turned on and off when they're needed, or have the gene completely turned off. It just doesn't work at all. And then comparing these plants, how they respond to PVY, to a non-modified Premier. And to knock it out is my main job. Emmerich developed the overexpressors. I'm developing the underexpressors. And we had two strategies setting out. The first one was CRISPR-Cas9. Probably heard about CRISPR. It was originally a bacterial immune system that bacteria used to recognize invading nucleic acids, things like viruses, transposons, and then chop them up and remember what they were so it can chop them up later. And so the most simple way to describe it is they have a guide piece of RNA that a part of it is complementary to whatever genetic element they want to knock out. And then a protein called Cas9 picks that up and then carries it, anneals it to the target, and then chops it up. And a number of things can happen, usually resulting in, or not usually, I shouldn't say that, but often resulting in a damaged gene, a gene that either has deletions, mutations, things that make it not work, or it's just, depending on how you design the guides, it can be chopped all the way out. And the best one I have so far is a line that has one allele completely truncated, so this one is not functional. And then, potato is tetraploid, so if you knock out one allele, you still have three that work. So that's not good enough to fully have a knocked out plant. So we have another strategy called microRNAs, or artificial microRNAs, which is based off of a similar mechanism that's actually natural in plants called RNA silencing, whereby these small hairpins of RNA that are complementary to certain genetic elements are expressed, picked up by some proteins, and then used to recognize transcripts of the target and to chop them up. It was originally discovered in petunias. They wanted to enhance the purple flower of a petunia, and what they ended up doing is turning off all the purple in the petunia. And so through this strategy, we're working on knocking out the expression of the target genes. And so the general strategy to get things into a plant, get new genetic material into a plant, making GMOs, transgenesis, another way to call it. First, you construct a plasmid program that you put into a bacteria called agrobacterium, that its natural life cycle, it places a piece of its DNA inside the genome of a plant, its host. And what we do is just replace what it will usually put in with what we want to put in. So all the bits and pieces that we need to make these hairpins, or the protein Cas9 in the guide. We put into a program, put into the bacteria, inoculate our plants with that bacteria, and then there are going to be single cells that have modified DNA now, the cells that got infected by the agrobacterium. And what we do is place those plants onto a kind of medium, a defined medium that has hormones that make the plant de-differentiate into kind of a cancerous mass of replicating cells that are identical. and if one of those masses received the new gene earlier on in this step, then when we redifferentiate it, we put it on a different media to make the dedifferentiated masses of cells turn into new plants. Ideally, these plants will have the new genes in every cell. And then we chop it off the callus, and then we root it in media. Here's some more pictures of that. They kind of look like broccolis, but these are just stem pieces of potato. So think of this, you just chop it like that, and then put it on the media. Well, inoculate it with agrobacterium first, and you can see little plants start to form. And so once we have these transgenic lines, we plant them out in the greenhouse, infect them with virus, essentially, and then observe development of symptoms. And then we infect them with multiple different strains of the virus and compare them to the non-modified control. I'm still working on the knockouts, the ones that reduce the expression of the gene, but we have the overexpressors already, and we have a few results. So one thing that we noticed with the ABC overexpressors is that they develop symptoms more quickly and more severely when inoculated with PVY-NTN compared to non-modified Premier. So you can see how this has seemingly healthy leaves, and these ones do not. They're kind of old plants, but you can sort of see what I'm talking about. And so working on trying to figure out and replicate and see what could be happening here. Why does having more ABC mean that the virus causes more symptoms? Is there more virus, or are just the symptoms being amplified? And then for the MYC overexpressors, we see a very clear phenotype just in the plant itself. So this is just normal Premier russet. It was mock inoculated, but there shouldn't be any virus in it, so it just looks fine. Versus this one, it's a MYC overexpressor, has a very clear phenotype. The plants don't grow as tall, the stems are more robust, you can feel the leaves, they feel differently, and they develop this kind of chlorosis. Like this wasn't infected with a virus, but it almost looks like it has mosaic, or some sort of chlorosis disease, but that's just what the plant looks like. So we're working on trying to figure out, or just get hints at to what having more MYC does to make this sort of symptom. And another interesting thing, there are more interesting things about it, it seems to be more tolerant of drought stress. If we have a really hot day and maybe don't water as quickly as we should, we notice that these plants seem to be more like turgid and healthy compared to the rest of them that are more wilted if they go without water. And another thing, in our first trial of these overexpressors with inoculation, this line specifically seemed to be basically immune to the virus. It didn't get any systematic infection with PVY-O, and it only got one systematic infection out of 6 plants with PVY-Wilga, versus 5 out of 6 for Premier in O and 6 out of 6 for Premier with Wilga. So we're interested in that, and we're interested to see if the underexpressors that should be ready within the next few weeks have a commensurate inverse response, or if there's something totally different. We have no idea. That's the beauty of science, to figure things out. And that's essentially it. I want to thank everyone. Oh, and I had an assistant mix up a Rubik's Cube for me, just so I could prove that I could do it. So feel free to go get more pizza or whatever while I'm doing it. But
Question for Max?
yeah, any questions? This should be fun. No? Okay.
What are your plans when you finish up?
What are my plans? I'm interested in moving back to the Midwest. There are lots of jobs for the sorts of things that I do in St. Louis, and I also have a bunch of family in St. Louis. So I'm interested in greenhouse tech jobs or laboratory tech jobs. Kind of the trajectory of my career has been, I want to be able to do kind of every step in the process, and maybe someday I'll focus in on one step. But for the time being, I'm kind of general at the moment. But yeah.
That's not easy to do at all.
If anyone wants to try, you could do it.
So now you are working on the RNA silencing or the CRISPR?
RNA silencing mostly. Because the CRISPR, it's going to look like we need tons and tons more plants to get an ideal knockout event, versus RNA silencing, it's more tried and true. So, yeah.
No more questions? I would like to encourage all the students to present. You see, we have an example of a very bright student in front of us. But I know we have our schedule full for 2019, but for 2020, it's open, and it would be great to hear from each of you about what you're doing here. Everybody is included, so just let us know, okay? Thank you very much, Mike, and thank you everybody for coming.
Thank you. The thing ball? Here you go. Oh, there it is. Okay, ball.